Hi everyone, my name is Camille Maumet and I'm from INRIA based in Rennes, France. I'm happy to be here today as part of this course on cultivating open science practices in academic research and culture. And I will discuss more specifically how we can share more research outputs and how we can do so in a manner that is fair. But before we get going into the how we can actually do that, I wanted to stop and reflect a little bit on why might we want to share more research products. So here on the left, you have a high-level representation of a brain imaging study. So we're going to acquire some data. So here I've put examples from MRI, but that would be true of any acquisition instrument, EEG, MEG, what have you. We get a bunch of data sets, and then we're going to have a first phase of data preparation where we remove artifacts, we put all the subjects in the same space and so on. We get some measures of interest that are here called the derived data, and then finally we're going to run a statistical analysis or machine learning algorithm in order to get our final results. And if we are happy with what we've done, we typically write a paper that describe really nicely all our experiments. So this is the view of the person doing the experiment, the researcher doing the experiment. But basically, what we're going to share with the community is only the paper. And the view from someone from the community to get access to this research product will only be the paper. And of course, this creates, leaves the researchers who's looking at the study often with many questions. So here I'm going to take examples from fMRI, but you could transpose basically to any research type. So an example of question could be, well, which release of SPM was actually used in that study? Sometimes it's going to be described really well in the paper, and sometimes this information can be missing. Or maybe you're wondering about which registration algorithm was selected, or how the brain mass was computed exactly, because that's important for the region you're studying, and so on. And basically, it's a lot of work when you want to build on someone else's work to restart from zero in order to build on the results. And this is very important in research because a lot of the time you're going to want to compare your results with what was published previously. And then even another, on top of the idea that it's a lot of work, sometimes the results you just can't reproduce. And when you end up in that spot, it's really, really hard to understand why this is happening. So you might wonder whether you understood the method correctly, or maybe that's just due to the data set, or maybe just a different computing platform, or there is another explanation. So starting up with just a paper when we want to reproduce results, and I'm sure that's an experience that a lot of you have lived already, is both a lot of work and can be really frustrating, because if we can't reproduce, it's really hard to know what's wrong. And so the idea of this talk, as you've realized, is to be able to share more than just that research paper so that others can build on our work. And as was said in the title, our focus here is to work with research outputs that are fair. So what do we mean by fair? Fair stands for findable, accessible, interoperable, and reusable. And we'll get into more details about what this means practically. But often when we hear about FAIR, we think technical solutions, standards, databases, good practices. And this is very true. Like, in order to implement FAIR, we need technical solutions. But before we get started, I want to emphasize that FAIR neuroimaging is also a way that leads us to a culture change, to a research that is more community-driven, because basically you're making more resources available to the scientific community, but you're also reusing existing resources, and that creates more links with different people that can lead to collaborations or new research projects or ideas that are exchanged and discussed. And all of this makes it a less isolated experience in research. So now we'll get to it. So how can we share more research products? And of course, I've mentioned the paper before and said that this was what we share with the research community. And I'm not going to cover this in details, but of course, for these papers to be accessible to everyone, you should consider using pre-printing system or post-printing system, just like such as bioRxiv, archive. In France, we are lucky to have a national archive that is called AL, and so on. So that's our typical research outputs. Now, what can we share other than that? Well, in the other talks, you've heard about working with large data sets. And one obvious research products that we might want to share is the raw data. So here we have our raw data, and there are a number of databases out there that will let us share that raw data. So OpenNeuro is an obvious one, but there are also alternatives, especially in Europe. So I'm thinking about the Dundas repository, which is only for the Dundas lab, but there is also Loris in Canada, Chanois in France, and so on. So we have repositories where we can go and put the data. So this makes those data sets findable. And now we're going back to our FAIR acronym. So F stands for findable. And basically what findable means is that I can search for open data set in each of these repositories. Obviously, this could be even better with a search across repositories, but at least we have some search functionality. Now, the next letter is A for accessible. So what do we mean by accessible? Accessible means that basically, on top of being able to say there is an open data set available somewhere, I can actually go and get this data as a researcher. And here, we typically, for raw data, have two approaches. Either you're going to have a direct download, and that's what you found on your Vault. So basically, findable and accessible are just both done together. Or you might have an extra step with, as a researcher, having to sign a data user agreement. And this is what we typically see in Europe, for instance, to comply with the GDPR, the General Regulation on Data Protection. And the data user agreement will ask, for example, the researcher who are downloading the data not to try to de-identify it, not to reshare the data without letting know the people who own the repository, and so on. So there are a number of rules that makes it possible to access the data and reuse it. So we've done findable, accessible. Then the next one was interoperable. So what do we actually mean by that? The question here is to be able to find a common language between different tools. So why do we need that? I mean, if you've worked with someone else's data, you're probably aware that the data itself, by itself, is often not enough to work with it. And I'm not talking here necessarily about sharing data with the research community. Maybe you've tried to reuse a data set from your neighbor in the lab. And so they share the data, and then it comes to you, and then you have a question because you're not sure whether, well, is that data from that subject? Or maybe there is a parameter that you need to run your analysis and so on. And there are typically many information that has to come with the data for the data to be just usable. And those are called metadata. And the standard is here to have a common encoding so that instead of having to ask each single person where this parameter is stored, we all agree together and we store them in standardized way. And so for raw data specifically, we're lucky to have BIDS. So BIDS started as an effort back in 2016 that was focusing mainly on, initially on MR, but it has extended to many other modalities, including PET, MEG, EEG, IEEG. And so it's a way to store the data for different participants with enough metadata about these data sets so that they can be reusable. And so this, by using, so going back to our question about interoperability, basically BIDS acts as a common language across tools because many different tools can understand that structure. And I've mentioned reuse already a few times, and what's nice about BIDS is that it was built with reusability in mind. So an example here is that amongst all the metadata that are included, there are the onsets for task fMRI. And basically, if you have a task fMRI dataset without this information, there is nothing much you can do. And so here we've run the full circle for raw data of having fair data sets. And I think that right now for many modalities, we have ways of sharing data set. Of course, there are many more considerations, including like consent forms for participants who are sharing raw data that I will not cover in this talk. So we've looked at raw data. We found that we can package them with bits, and that there are a number of repositories where we can share them. Now, what we've done with raw data, we can also think about for results. Because yes, well, in paper, we share some information about our results, but it's usually pretty limited. So you might have maybe a few slices that describe your main findings, but it's never as good as 3D volume of a brain, or as your full signal, that is your results. So basically, the same question here arises for results data. Can we share results data in a fair way? And here I'm focusing on task fMRI. And so for task fMRI, we are lucky to have a repository which is called Neurovault, where anyone can go and upload their statistic maps, which are the results of fMRI study. So here we are basically good with with findable and accessible. So we have this repository where we can search for data sets. And typically data sets are available as direct download. And because often statistic maps are average across a group of subjects, it's a bit easier than sharing raw data and having to have data user agreement to respect the privacy of the participants. So now we can ask ourselves again, do we have a way to make this interoperable and reusable? So do we have enough information with our data sets so that they can be reused by other people? And here I'd like to talk about another effort, which is called NIDM results, where the goal was to have a model to represent the results of task fMRI studies. I'm not going to get into the details of all the pieces that are included in there, but just to say this includes a few maps first. So the statistic maps, but also the contrast maps, which can be important for reuse, as we'll see later on, the parameter estimates, but also information about how was the thresholding done, for instance, or which contrast specifically was used, and so on. And again, this export to NIDM results is directly available in SPM and FSL and can be used to import data in Neurovault. So here we have really our common language that lets us go from one software package to a repository or another software package. And the goal of NIDM results was to be able to enable meta-analysis. So the only meta-analysis we can do with the information that is available in papers are coordinate-based meta-analysis. So meta-analysis that combine information about the peaks. But with NIDM results, because we share more information, we share complete maps of the brain, results maps of the brain, we can go slowly towards image-based meta-analysis. So here, again, we have the full circle of findable, accessible, interoperable, and reusable data set for task-based fMRI results. So we've looked at raw data, we've looked at results, and here we have a way to package them again with the standard and then dedicated repository. We could ask ourselves about derived data, basically what is in the middle. Might we want to package these and share there as well? And here it's a bit more complex, as you can imagine, because basically derived data can be very, very different. So there is probably often no standard way to decide what's a derived data set. And so here, so far, what I encourage people is to look into general purpose repositories. So we have Nitric, we have Zenodo that basically accept any type of data set. So of course, you're going to have less information about these data sets than in a dedicated repository, but at least if you have a derived data set that you think is important to share with the community, that's one way of doing so. So here again, data sets can be searched and accessed through these two repositories. Although, as I said, the search will be less specialized than if you had a dedicated repository. Now, going into the question of interoperability and reusability, in order to be able to do that, you really need a standard that is going to describe this data with their metadata. And here, of course, it's going to depend on the actual data set you want to focus on. And there is no single answer, but I would at least encourage you to look into two directions. First, try and find existing standards and databases, because if they exist for your particular data set, then that's probably the easiest thing to do. And this will maximize reusability for other users. And so the fair sharing database is here to describe different standards, databases, policies with different types of data. So that's a good entry point to at least try and search to see if you can find a pointer to a standard or repository that you might want to use. But if it turns out that there is no standard available, you might be the one of the best person as someone who knows this type of data to help develop the next standards. And here I would encourage you to look into BITS derivatives. So just like BITS started with raw data, but is now growing into describing many different types of data, including derived data sets. And there are a number of BEP or BITS extension proposals that focus on different aspects of the data. So I've put a few of them here, including structural preprocessing derivatives, functional preprocessing derivatives, diffusion weighted images derivatives, and so on. And so you might want to go there and see where the standard is at and maybe contribute. One of these BITS extension proposals that I would like to describe a bit more is BITS provenance. So the goal of BITS provenance is to have a model that is agnostic to any type of data. So instead of trying and representing maybe just the results of, I don't know, fMRI preprocessing, here instead we focus on describing all the steps that led to a data set. So you might be able to say this data set was obtained by running this tool with that command specifically and this set of parameters. And after we ran another algorithm with these parameters, and that took as an input the outputs of the previous steps, and so on. And you can chain processing and basically describe a full pipeline. So that's the goal of BITS provenance, which is currently ongoing, and seeking community feedback. So I would encourage anyone interested to have a look as well. So we've looked into derived data. And probably here, so I'm going to link to more general repositories, and I would say you'd have to look specifically for your particular use case. And this is true for results as well. So I've described task-based fMRI results, but for your particular type of results, there might be no standards out there. And then the best thing to do is just to try and upload on a generic repository. So we've looked at many different types of data sets, but there is another piece of the brain imaging experiment that we haven't covered so far. And I'm thinking here about these two steps, so data preparation, statistical analysis, that are typically described as a piece of code. And here, I'm not going to get into many details, so you've seen how to build reproducible pipelines, but sharing analysis code is a different space and is something different. So you have different requirements for sharing code comparing to sharing data sets. If we think about it, code is often stored, like if you're using version control, it's often already stored in online repositories. This can be GitHub or GitLab. Code has typically many versions. So of course you can have versions on data sets as well, but for codes you could think of each commit being a different version of your code. I'd like to emphasize here that just like for data sets, we use repositories that are here to keep the data in the long term. We need to think about long-term preservation for code. Because basically, if you just put your code on a GitLab or on GitHub and you don't think about it more, those code might not be available in 10 years from now. And so there are dedicated platforms such as Software Heritage and Zenodo that let you store the code and make sure that it will stay in the long term. Also, when we think about code, it might be good to adopt a standardized structure. There are a couple out there. I've put one here, where basically if each project, if your project has the same structure, it's much easier for first people in your labs, but also people outside of your lab to understand what's going on in the code and reuse it. And of course, having a good README, I haven't put code of conduct, which is of course very important. So here we have a view, again, with like, we've seen how to share raw data, how to share results, derived data, and our code here. There is one aspect that I've completely overlooked in this code, which is sharing information about the acquisition. And here again, it's a really, really, really big world. So you might want to share, for instance, the images that were used in the DMR machine while the participants was the acquisition while running. Or you might want to share the instructions that were given to your participants, because we know that different set of instructions can have an impact on the actual results. You might want, of course, to share details about your imaging sequence and so on. So there is a big space here that I haven't covered, but many different research outputs that could also be shared. So just to conclude, a few last words. So are you ready to go with us and share more research outputs? So a few rules here. We've seen many different types of research outputs and different ways of sharing them. But what's really important to keep in mind here, I tried to give a comprehensive picture, which is still not complete. But the important bit here to remind is that you can choose. Pick one or two research outputs, the one that are easiest for you, or the one that you care the most about. It could be just your code, because sometimes maybe in your lab it's not possible to share raw data because consent forms haven't been updated. Well, focus on what's easy for you. Start with the code maybe, and then later on you can go to other research outputs. Even within, if you've decided to share code or results, you don't have to share it all. You can share just pieces. Maybe you can share the code to build your figures or part of your results. If you think data sets in particular are very valuable, I'm thinking here about maybe manually delineated regions. This is extremely valuable. Maybe focus on sharing those rather than the full spectrum. When you feel more comfortable, I would encourage you to consider working open all the way. So opening up first makes it easier to share later on. So I'm thinking about code, obviously here, where you can start developing in an open repository, and then you don't have to ask yourself, is the code like, you don't have these steps where you have to check if your code is good enough to be shared. It's open, people know it's work in progress, and the code is gonna get better and better and better, and at some point it will be linked to the paper and it will be open all the way. And of course, I can only encourage you to connect with the community, help advance fair research on the research outputs you care about and you love. So if I have just one sentence to finish, pick what you like the best. You don't have to do it all. Thank you so much for your attention, and I'd be happy to answer any questions.